Hi, I'm Jordan. This is the last lab of the year, lab 12, color, vision, and perception. Alright, so for the first part of this lab, we're given a slit with a 20mm inch pupil, and we're supposed to hold it at arm's length and see what our dominant eye is. I noticed the best way to do this is to hold it kind of so the center of the inch pupil bisects your face, and I'll hold it like this, and I'll look at something, and I'll slowly close it till it's 20mm, and I'll switch eyes, left and right. Whichever eye the image plane shifts, that is not your dominant eye. So whenever it remains the same, that is your dominant eye. Turns out that my right eye is dominant. I'm also right-handed. Okay, so for the next part of this test, we're determining our blind spot of our eye. And so what we're going to do is we're going to hold this at arm's length, this paper with 2 dots on it. It's called the 2-dot test. And we're going to see which dot disappears as we move it closer to our face with one eye covered. So I'll cover my left eye and I'll focus on the left or the right dot. I'm going to slowly move towards my face and wait to the point where I can no longer see the dot on the left, and it's here, and then it's going to reappear one more time right here. And from that we can determine the field of view of our blind spot. It turns out that for me it's roughly 10 degrees. Alright, so for the next part of the test, we're determining our field of view for each eye. And so we've done this a million times, we know how to do it. We take a known distance, a ruler stick, and then move it a known distance away, and we wait till it fills the field of view of our eye. So we slowly move it, measure it. We know this is a yard. This is roughly 31 centimeters. I had a field of view in both eyes roughly the same, 112 degrees. And for the next part of the lab, after that, we have a visual acuity test. So we walk over here, we can see the Snelling eye chart. And the way it's calibrated is at from 20 feet away. This line right here, line 8, that's considered 20/20 vision because these letters will subtend 1 arc minute. And so I'll walk over here, and the way this works is you basically just stand here 20 feet away. Now line 8 subtends 1 arc minute, read them off, and from that you can determine which line you can read the most of, and that is your visual acuity. And that was it. Thank you.
Hi, I'm Garrett, and another thing we did here with the human visual system is to see the maximum frequency that we can detect. And we did this experiment with a whole bunch of different conditions. Essentially, we have a function generator here. We can vary the frequency and the voltage amplitude applied to the wave. So we looked at 3 different LEDs. There's a red one, a green one, and an orange one. The red one turned out to be, we had the lowest at about 32 hertz. The frequency sort of washed out. Green was a little bit higher at 43 and 44, but what I noticed is with the orange, the highest frequency that we were able to detect, Jordan and I, was about 48 hertz, and that was at 3 volts. An interesting thing to note is that when we bumped it up to 4 volts, that actually decreased, which sort of is counterintuitive. You would think that 4 volts you have a greater amplitude, greater signal-to-noise ratio, greater contrast. But that wasn't the case. Another interesting thing to note is that when this is put up against the metal table, you can see the frequency off of the reflection, but not necessarily off of the bulb itself at high frequencies. And I thought that that could be maybe a function of reflected light is polarized, and we may be sensitive to higher frequency light that's polarized. Or perhaps it's just going over a greater area. So I tested it on a paper too, and it seems, you know, when it's dispersed off of paper, it's also a greater area, and it's also a little bit easier to determine the spatial frequency for that. Sensitivity chart. Essentially, you have different spatial frequencies: 2, 4, 8, 16, all the way up to 64, and then you decrease the contrast until it just washes out. And the interesting thing to note here is that at very high contrasts, or at very high spatial frequencies and at very low spatial frequencies, you need a lot more contrast than you do for, say, 16 or 32. It's much easier to determine contrast when the spatial frequency is within that range. So it sort of follows this U pattern. It's not linear. That's all I have to say about that.
Hi, I'm Ryan. This is our last lab. I'm going to talk about the color limits, the spectral limits of the eye. So here we have a color spectrometer where we can test our limits. So we have our incandescent source. It's going to come in through this adjustable slit. We can use this to adjust the width of the slit. It's going to bounce off of this mirror. I don't know if you can see it. It'll come to this mirror. bounce off our grating, and our grating can be adjusted using this. If I go fast, you can see it rotate a little bit, so it changes the angle, and the grating reflects to this mirror, and then it comes out here where we'll be able to see the colors, the spectral lines. So what we do is we cover it up. We want to use something to keep us from touching the light, and we can adjust this and find where we can and can't see the color, the upper and lower limits. And we found that for this lab, around 765 was our upper limit and around 380 was our lower limit. Alright, and then for the last part of the lab, we dealt with magic images, optical illusions, stereograms, etc. I'm going to focus on stereograms and magic eye images, talk about how they work. So for a stereogram to work, the concept is that your left eye and your right eye both see different images, and the pasting of those two images together by your brain creates a 3D image with depth and clarity, contrast, etc. So in order to make a stereographic image, you have to give your right eye and your left eye something different to look at, and then put it together into one image. So an example of that is right here. I constructed a very basic stereographic image. So we have a left eye image and a right eye image, and if you cross the two together, you'll get a 3D cube popping. So in a sec, I'll ask you to pause the video and try it yourself. It does work. The reason that it works is that the perspective is slightly shifted. This is a cube drawn with a certain slope. It's a y equals x slope. And this is a box drawn with one more square slope. So it's a 2/3 slope. So we have 2 units and 3 units, and this has 2 units, 2 units. So that slight shift in perspective allows you to see a 3D image. So go ahead and pause.